So I'm junior faculty in the Division of Nephrology, and I've been using metabolomics for the last 6 years and learned a lot by trial and error and a lot of oops, I should have thought of that. So I'm here partly to share my knowledge and experience with you guys. Some of this might be very basic, but believe me, the reason they are on the slides is because somebody has forgotten it in the past or has overlooked it in the past, and therefore it's something I would like to stress. And quite a bit of these content is repetitive for a reason because of the importance of it. So here we go. So we are here at this point where you have an idea about an experiment, you have a hypothesis, you want to test, you have a question that needs answered. At this point, consultation with the core is highly recommended because there are factors sometimes that you do not think of that the core consultation people could pick up and help you way before your experiment actually gets done and avoid any future mishaps. And at this point, you have to carefully design your study so that you're taking into account all the specifics that metabolomics puts on your existing study design. Also, to think a little bit about how are you going to collect the samples, how are you going to store it, because this has huge impacts in metabolomics experiments. So this talk will cover how you will answer your biological questions with metabolomics, what are the factors to consider in designing a study, and how to account for all the variability that I'll be talking about. So we are all now familiar with these factors of systems biology, genomics, proteomics, transcriptomics, and today we are talking about metabolomics. All of these omics studies describe the phenotype that you're studying, which is your ultimate goal to figure out why, how, and how come. So, but all these omics platforms are influenced by environment that's surrounding them, especially diet, age, lifestyle, drugs, and disease. Less so by the genome platform because those are things you inherit. But the transcriptome and the proteome are also affected by these factors, but more so in a longer period of time. Metabolomics responds very quickly to these changes in minutes, sometimes in hours. Therefore, making small changes in your study design, huge impacts on your results. So that's why we're going to pay some attention to these factors. So today I'll talk about the different model systems that can be used, the different biological materials that we can gather from these systems, how to design a good study with good power to get your answers, and also talk to you about some examples how variabilities affect the metabolome and give you some sample collection strategies to avoid these variabilities. So model systems, the fundamental model system is a cell study. It's a very controlled platform because you're growing cells usually of one type. You can either measure the endometabolome of the cell that you're growing, or you can measure the exometabolome, which is the extracellular milieu, by collecting the culture media. These are very reproducible because the factors that control the growth and metabolism of these cells are totally under your control. And therefore here you don't need very large sample size, and quite reproducible metabolome can be achieved by this. We also have the ability here to follow the flux of the metabolites using tracer compounds. As you can see here, you can load in isotope labeled tracers in the culture media and follow the metabolites across the pathways. And as the label passes through the pathways, various isotope labels get enriched in the pathways, and those can be formed. And this is the basis of fluxomics that will be talked about later. Animal studies are also great platforms to answer your questions. Here, the inter-individual variations are reduced because usually the mice are from a single genetic background and they have been controlled environment in your vivarium. You're feeding the diet that you want, and therefore small sample sizes are enough. And here you have the opportunity of simultaneously harvesting different biological materials, including fluids and tissue. Therefore, you get an overall picture of the metabolome of the study. Humans come from various genetic backgrounds and live in various environmental situations. Therefore, the individual variability in humans is quite high. Therefore, because of this, you need large sample sizes to characterize these and tone down these biological variability. The good thing about the human metabolome is more than 4,000 metabolites have been characterized and studied, and target ranges have been well published. There's a huge consortium called the Human Metabolome that has published these values. Therefore, there are things that you can refer back to. So that's a great advantage with the human metabolome. The biological materials that we can collect, I kind of alluded to it already, cells. The amount you need is around 10 million cells, and culture media can be studied for the extracellular milieu, plasma, serum, urine, of course, saliva, CSF, pleural fluid, ascites fluid, tissues of various kinds depending on your question in hand, skin, nerve, organs. Of late, for microbiome changes, fecal material can also be studied. So these are varied things that can answer your questions either in individual manner or in cohort, so you can get the whole picture. So what type of studies are conducive to metabolomics? I'll start with a case control study. In a case control study, you have the opportunity to well match the control cohort to the case cohort. Therefore, toning down the discriminating factors that discriminate the metabolites, except the specific question at hand. So if you're very clear on the question at hand, you make sure you match your case and control with all other factors that's involved. Therefore, you can have a very clear pattern of the differences. These have usually excellent correlation to the disease patterns, can open up biological pathways and biomarkers. They can be easily powered to detect differences if they are matched really well. But they are, of course, prone to biases because it doesn't prove causality, and there is some very rapid changes in study design that can cause changes in the answers you get, causing big artifacts. So that is to be kept in mind. In interventional or time series studies, you have an opportunity to do a before, after, either an intervention or change in phenotype. This gives us an idea about the stability of the metabolites as well. If you have a longitudinal study that you're following over many years, you have an opportunity to see whether you're able to measure a metabolite that predicts something over time. Is that reliable? So if you have many time points, that's a good thing to figure out. The other thing you can do is, because you're measuring the same study subject before, after, you can kind of control the inter-individual variability. Crossover studies are also good study designs because the patient can be their own controls. Therefore, you're taking away the inter-individual variations, and because of the many time points usually in these studies, you get reproducibility checked as well. So we come down to sample size. What is the appropriate sample size to get an adequate power? This is a common question. And the reality is, with all metabolomics experiments, including the targeted metabolomics experiment, you measure a lot of variables. When you increase the amount of variables that you measure, you increase the chance of having a false discovery rate, which is, in other words, you're increasing the type 1 error in the experiment. So you're rejecting the null hypothesis when it's true. In order to account for this issue, we have to choose a stringent cutoff with a lot of statistical techniques. The stringent criteria translates to you as more sample size. So if you don't do an adequately powered study, because of the number of variables and the stringent cutoff that needs to be applied, you will lose data that might be in the background if you do not power your study well. So one strategy to get around this issue is to get an idea of the effect size, and we typically suggest that you have a small pilot experiment designed so that you can know what the effect size of your intervention or your study question is. Therefore, using that effect size to design a larger confirmatory validation cohort or validation experiment that can get you the answers that you want. Other issue with the metabolome is the biological variability. This is especially true in humans, that we are all not the same. You ideally want to look like this, separate out two groups like this, but the reality is inter-individual variation is so high. Even with intra-individual, as in over time, you will see that the variation is really high. To account for this as well, your sample size needs to be higher. Higher is better. So to account for the short-term and long-term variability, powering your sample size is really important. The other thing to consider in metabolomics experiments is to data normalize, as in you are collecting biological materials. How will you normalize a sample A to sample B? Sometimes it's really clear. For example, cell number in cell experiments, volume in media, and biological fluids like plasma. Sometimes you are doing fluxomics and you're trying to normalize two subjects together. You might use key metabolites in the plasma like glucose. Urine, I'm a nephrologist. In urine, we know for sure the concentration effects are really huge between subjects and even within a subject depending on your hydration. So you can use metabolites like creatinine, which are fairly stable over time, or the osmolarity of the urine to compare sample A to sample B. Other things to use in, for example, is in tissue is use protein concentration. Dry weight of the tissue, we use that in fecal samples because to normalize between two samples. If all these methods have not been thought of, you've started the experiment, we can also normalize using the total ion chromatogram, which is the output of the chromatographic separation method. So I'm going to give you a few examples of why I keep stressing the study design aspects of this, and then we can go into the study sample collection aspects of metabolomics. So all these things matter in metabolomics. For example, in this study here, this is a study, a cell culture experiment with myeloma cells. And here you can see as time goes on, the amount of lactate goes up in this experiment compared to the amount of glucose that goes down. So depending on where you sample this cell culture experiment, you're going to get a different ratio of glucose to lactate. I'm just illustrating glucose and lactate here, but many metabolites change rapidly in a cell culture experiment. So standardizing your time of harvest of these cells is really, really important. Also, seeding the same amount of cells, catching them in the same cell phase, passage number, these are things to consider. Growth conditions, if you change the temperature midway through the experiment or change the ambient atmosphere, these will have huge changes to metabolism of cells. Also to think about is what is in your culture media, because if you're studying a fluxomics with labeled glucose, you don't want glucose to be in your culture media. So those are things you need to figure out ideally through consultation with the core. And if you change culture media halfway through the experiment and one had growth factors, the other didn't, those are going to give you huge changes in your answers. Timing. Here we have plasma samples that we looked for polyunsaturated fatty acid. You can see as time goes on, these polyunsaturated fatty acid levels go down and go up. And at a 24-hour mark, you can see this is really a circadian rhythm here. So here, choosing the time of sampling this experiment is really, really important because you can capture a different metabolome. And if there is variance between your sample collection of your entire cohort, you're going to have some artifacts that are going to influence your answers. So these are things to consider. It is not just time, it is also what activity, what fasting state you are on, whether you had a diet recently, all these things influence metabolites, some metabolites more than the other. Here is a study where they measured the mean carnitine and free carnitine, which is in the red, and the green is the acyl carnitine levels. The first part is fasting time, so over time, 24 hours, you can see the changes in the carnitines. This is a liquid diet with lipids, changes. Glucose tolerance test, you can see obviously the changes. This physical activity test is making the cycle for an hour. You can see the differences in the carnitine levels. And this is cold stress. You can see dipping your hand in 3 minutes in cold water makes a change in your carnitine levels. So metabolites can be very sensitive to these kinds of changes in the environment, and you need to account for this in your study design. This is another example of diet, and this is urinary metabolites, with diet 1 being saturated, diet 2 being monounsaturated, and diet 3 being polyunsaturated. You can see in this PCA plot where these diets separate out very quickly. This is in a 4 hours time period. So what the patient has eaten, if this is your intervention, that's great, that's a successful experiment. If this is not your experiment, then it adds to the biological variability. And what about collection strategies? How does these affect? So choosing the right tubes, whether it's serum or plasma. For example, in this study, comparing plasma with albumin blanks, plasma and serum, you can see the differences in the peaks of these untargeted chromatogram here. You can see that plasma looks different than serum. Though many of these metabolites do correlate, there are some that do not correlate, and these need to be accounted. So halfway through your study, do not switch from serum to plasma samples. If your metabolites are those that do not correlate, you will have different answers. So this speaks again to the word we're going to constantly hear today is consistency in your sample design and collection method. Also, sample processing. Are you washing with a buffer that's different? Is metabolism quenched at the exact time, every cell plate? Are you scraping? Are you digesting? What about centrifugation speeds? Because cell lysis can occur, changing the metabolites in your pellet. Hemolysis with the biological fluids like serum and plasma could also change a lot of metabolites. In this little figure here, this is a paper by Lorenz from the core. Here it shows the differences between storing the cells at liquid nitrogen quenching, -80 and 0. You can see the varied metabolite differences of the TCA cycle. So all these small changes in your protocol might make huge changes to your outcome. This is another example of collection strategies. This is urine left out on the bench. You can see this is a spectrum, NMR spectrum of spoiled urine. This is the normal urine before it was left out for 4 hours. You can see that new peaks start appearing. So you want to ideally store urine immediately on ice and add sodium azide to prevent bacterial growth. About temperature. If you leave samples on the in room temperature, here they're showing illustrating hypoxanthine and sphingosine 1-phosphate. Over time, these metabolites increase, and some of the metabolites might decrease in concentration as well. So standardizing how you're going to store during processing and when and how you're going to store them in -80 also makes huge changes. So process on ice, limit the processing time, transport on dry ice, and store as soon as possible in -80. And also freeze-thaw cycles. Have you stored your samples in a huge aliquot and you are constantly thawing them to get smaller aliquots? If some of your samples have been thawed a couple times and some are many more, you will have big differences in your metabolites. So the strategies to prevent variability are very simple, but really need to be thought of ahead of time. You time the sample collection in relation to a meal, ideally fasting, or document physical activity, take careful diet and medication history, document all sample related information, whether they are obtained in batches, are they stored in different freezers, did the temperature change. Standardize these collection strategy and keep the samples frozen and limit freeze-thaw cycles. So to conclude this part of my talk, a good metabolomic study design should have a careful design about the variability that we talked about and document all the changes and sample collection strategies and standardize them and store in -80. Two words you will keep on hearing throughout my talk is consistency and consultation. Consult before you start designing your experiment and consistency throughout your sample cohort. Now we can move on to talk about specific sample collection strategies because I showed you examples of variability, how to prevent them specific to the biological material you're trying to obtain. So as we spoke earlier, we have different biofluids and cell culture and tissues that we can obtain. If you go to our MRC Square website, you can see that under project requests, you can see this website, web page come up, and there is a link to the PDF. I'm told as well that you have it in your box folder. This opens up a PDF that shows you detailed guidelines how to collect cells, how to collect tissue, specific attention to metabolite panels as well. I will briefly go through an overview of these collection strategies and answer specific questions if you guys have any. So cell culture, you want to determine which is the best condition at which you want to have the experiment end. So decide when you want to harvest the cells, and each sample plate should have same amount of cells. So either count while seeding or at the time of harvest you count your cells. And you want to, after a quick wash, freeze with liquid nitrogen and store in -80 before the samples get to the core. You want to stagger the time of collection. This seems silly, but cells have rapid metabolism rates, but you want to stagger the start and stop in a sense that by the time you finish one cell, the one cell plate, the other cell plate doesn't have a larger time of metabolism going on. So you want to stagger them, allow time for the rinse and quench of each individual plate. Do you want to know what's in your media type and what is the nutrients in the media type, and keep to one batch of the media as well. And we highly recommend that you store a little bit of the media for analysis. We run into issues, we can always refer back to them. For blood, plasma, and serum, as I said, there are big differences, not only between plasma and serum. And here I'm showing you difference between EDTA, citrate, and lithium separated plasma. You can see that the EDTA plasma is our preferred choice, but you can see that the lithium has a lot more other peaks that come from the collection tube itself. It doesn't matter if you've already collected sample in the other methods, as long as your entire cohort is in the same tube. I think that's something we can work around with. Also, aliquot preferably in chilled tubes. You can see here the effect of freeze thawing. This is specifically carnitine measured. This is fresh sample that has this much carnitine. 1 freeze thaw cycle, 2 freeze thaw cycle, 4 freeze thaw cycle. You can see how the level of carnitine keeps going down. So these are things you need to think about, and a little bit of OCD in this actually will pay you in the long run. So consistency is the key, and it's probably quite related to success in metabolomics experiments. Biofluids, you want to snap freeze in liquid nitrogen and store in -80 and transport on ice. Here again, I'm showing you an example of plasma, arginine here. On room temperature, after 24 hours, you can see the arginine levels drop. If you keep on a cool pack for 3 hours, 6 hours, 24 hours, again, time of processing, time and lower temperature than -80 all have influences on your metabolites. How about hemolysis? You want to avoid hemolysis ideally by grading them and weeding out those samples. If your samples are precious, you want to go ahead with using them. You have to keep in mind that there are huge differences with hemolysis. This is about 69 metabolites that are fold change different from no hemolysis here, moderate hemolysis, strong hemolysis. Most of them are lysophosphatidyl-cholines, but there are other metabolites that change as well. So you want to limit processing time and hemolysis. And if you do proceed with hemolytic specimens, please document which ones they are, so that when we see an outlier in the analysis, we can attribute them to those causes and weed them out in the analysis. Urine collections, again, preferably you want a 24-hour urine collection that captures a change in the metabolome over the entire time and minimizes short-term changes in urine metabolites. At the very least, you want a fresh first morning urine. You want a midstream clean catch, and you want to store the collecting urine over ice or in a refrigerator while it's collecting. In animal studies, you want to use a metabolic cage and try to get as clean as a specimen as possible, which collects on ice. Here again, I'm showing the difference between the time of collection. This is urine on a PCA plot, collected 24-hour pooled and 4-hour pool. You can see that they all separate well. Over 24 hours, the samples become more tight. Here, the closed circles are collected on ice and the open circles are left on room temperature. You can again see the differences between the samples, especially in the 4-hour pool. So keep them on ice on collection, and the longer the collection period, the better. When you process urine, you want to add a little bit of sodium azide, which prevents the bacterial growth. I showed you a slide earlier why bacterial growth is important. You want to centrifuge on a standardized spin rate and store in 4 degrees throughout. You want to remove the pellet and don't go very close to the pellet while you remove the urine so that you don't get contaminants. Aliquot and store at -80 and transport on dry ice. Also, you want to know when you sample the urine, especially around diet and medication changes. Here I'm showing an example of urines from immediately after taking, this is normal, this is Tylenol, this is ibuprofen. You can see the changes after taking the medications immediately. This is 2 hours after taking medication. So you want to document a good diet and medication history and the timing related to your collection of samples. Ideally, you want to make a big form for your patients and ask them to fill those things out. For tissues, for all solid tissue, biopsy materials from animal study, usually we need around 50 to 100 milligrams. So you want to snap freeze them immediately. If you are planning to dissect out a certain part of a sample, please do so quickly on ice before snap freezing them. Typically, you want to consult with the core for little minute specifications that are necessary when you're collecting tissues. Store on -80 and ship on dry ice. And this is something, again, even tissue samples can change with the time, with diurnal variation, with the diets, with medication. Here I'm showing about 10 mice liver tissue here. And this is a light-dark cycle. This is starting of light time, and this is dark time for these mice. You can see that the liver tissue, the peaking of different lipid species occurs at different times. The orange ones are the triglycerides, and the green ones are the phosphatidylcholine. So you can see, depending on what metabolite you're looking, and the light-dark cycle for your mice, you can be sampling different portions of the metabolome. So one way to get around this is to standardize the time of collection of these tissues, which means that you can't sacrifice 100 mice in one day. You have to plan it out and document when it was done and be consistent with the same person doing it. If not, write detailed protocols so that people can follow the same thing. For animal studies, we do recommend that you perfuse with the PBS so that we get the blood out of the tissue. Because the metabolic concentration in the blood is really high, sometimes the differences you could be seeing could be a reflection of the blood circulation. So you want to remove blood as much as possible. We have a very good protocol of doing that. You can talk to us about that. So perfuse out the blood so you remove that aspect from your tissues. As I said again, dissect before freezing. And then general housekeeping tips. You want to label your samples with permanent marker. We have had a lot of mishaps with the labels falling off and not knowing which sample is which sample. Document, document, document. Because as I said, so much variability can be only accounted with careful documentation. Because you can go back and see outliers that we can account for because of batch effects, freezer changes, freeze-thaw cycles. If you're able to document all these things, it could be easier on the statistician to work around your results. Document the exact collection, the site, time of sampling. Please do write, especially for human studies that are done with large sample numbers and many clinical coordinators working on the same thing. Please get down to the nitty-gritty and document every single thing so that everybody follows the same protocol and documents the changes so that we can go back and weed things out. And consider your randomization during collection as well. You do not want to collect all your control in one day, all your cases another day, because you could have daily variations in sample correction that could cause artifacts in your study. Therefore, you want to randomize your groups. You want to do a little bit of cases and controls in one day so that you are spreading out the variability and getting a more clearer answer. And as I said before, consistency is key. Consultation is highly advised. We don't want you to spend a lot of money asking important questions and not thought of small things. These are very small things, but could have huge effects on your answers. Once again, apply for consistency and consultation. I'm showing you data here from rats that were sampled from the aorta and the jugular vein. You can see that using the PCA, we are able to separate out the sites of sampling as opposed to the individual animals. So the animals are separating out based on the venous or arterial blood. So not only sampling site, sampling time, change in diet just before your experiment will all be useful in the long run. So for all the people who have already finished your studies and thinking of running metabolomics experiments, do not despair. We have ways to work around it. You want to sit down and consult with us first and see what you have done, what kind of samples you have, what is the question you want to answer, so that we can come together and find a solution to the problem. Typically, you want to look to literature on what's out there and see how if your metabolite of interest, for example, doesn't change with diet as much, maybe a non-fasting sample could be still a good sample to work on. They are, as Chuck mentioned earlier in the talk, they are things that get challenged when they are in the non-fasting state. And if your experiment is asking that question, a non-fasting sample might be the ideal thing for you. So consultation is really ideal, and look to literature on what's out there. Also, I suggest running a pilot experiment. Not only will you know the effect size, you will know how much sample quantity and quality that you have. So that will save a lot of heartache, and it's great for use in grants as well. Please do plan a pilot experiment and power your ultimate cohort as a validation cohort with good power, knowing the effect size. Compare data analysis with sample information. This is really useful when I talked about the OCD of documenting everything. We have seen batch effects from freezer origin, batch effects from hemolysis. So these things, if you have the information, we are able to tease out in the data analysis aspect. We can also, using statistics, attempt statistical fixes to these variations if we know the details of the experiment. So this is the reason why you should document all the details, be consistent, and please do consult with the core. These are the references I used, and I will stop here to answer questions. Yes.
So I have two questions. Firstly, for the animal study, as you said, the life cycles and fat status can affect it a lot. So do you have recommendation on how do we decide the sampling time point?
So this is, there is no one answer for all these things, because as I said, there's literature out there on these things, thankfully. So if you're looking at phosphatidylcholins, you want to time exactly when they peak, if that's the question in your experiment. If you're looking at metabolites that vary with diet, you want to fast your animals for a certain time and then do it. So it depends on your metabolite in question. But as far as, if you have an untargeted approach and you're looking for changes, as long as you consistently sample at the same time, I think you'll get uniform answers. Instead of changing the sampling time, if you always sample, say 4 hours after the light cycle starts, it will be consistent throughout the experiment, and we have eliminated that variability out.
Okay, so if we don't really know what we are looking for, is semi-fasting recommended? Like when the light turn on, we fast it for a short period, like 4 hours.
Yeah, in our lab we do 2 hours. So
2 hours?
Yeah.
Okay. And second question, so at the data analysis part, if you find any artificial effect, for example, the fridge, as you said, so how do you correct for it?
One thing is to eliminate that sample out of your analysis, because this is a freezer effect. And if it's a whole batch of samples, I think they will address it later on in the workshop, how they analyze those samples. They add a correction factor to it usually. But I'm not a great statistician. I'll let the statisticians later in the workshop answer that. But they are fixes, not to worry.
Okay, look forward to it. Yeah. Thanks.
Yes.
For the tissues, can you do fixed tissues?
Usually not. It's fresh tissues that we use. I guess Maureen had something to add.
Should use the rules. We have on a couple of occasions, but it takes quite a bit of work to try to eliminate any effects.
Okay, it's doable. Okay. Thank you.
Oh, sorry. It's been done time and time again. It adds layers of evidence to your question, and it depends on the metabolites because what you're studying, the question you're asking, but it's been done many, many times. So yes, there is transfer of the same metabolomics from different model systems, and then integrating the results has been done.